Ladies and gentlemen in the audience here and online at home, wherever you may be, welcome to session 2, day 3, where we are talking. I have 4 presentations lined up for you on biotechnology, synthetic biology, and cell biology. We have a lineup of Marcel Egli, of Nils Nicolai, of Paolo Annibal, and Claudia Pacelli, who will be talking to you about on a journey through lower back pain to radiobiological characteristics of multipotent stem cells, looking at altered signaling in various cells, and finally some synthetic biology. So with that, I introduce to you Marcel Egli, who is fighting lower back pain of astronauts. Thank you, Marcel.
Thank you very much. Thank you very much, Ian. So, right. Yes, now. Okay, hello everybody, and welcome to the session. So as you've heard, my name is Marcel Egli. I'm the head of the Institute of Medical Engineering and the Space Biology Group at Lucerne, University of Applied Science, and the University of Zurich. So it's a great pleasure for me being here and actually introduce you to low back pain. And also thank you very much for inviting me. It's a great pleasure. Low back pain. I think that the study itself, it's an ongoing study, so I don't have final results that I can present. However, I think that the approach that we have used for this study could be quite interesting for you and maybe gives you some ideas on how you could do it. I will show you how we are doing things. So low back pain, this is a study that I'm doing together with Jan Swaneborg. He's from the University of Zurich. Well, before I'm going into the details, I would like to introduce you to low back pain in general. Well, low back pain is something that we are all suffering sooner or later, unfortunately. So we feel, pain in the lower back, and it's really hurting. And as you can see, almost all of us will suffer sooner or later under that. And apparently 20%, a little bit more than 20%, will actually develop a chronic pain in the lower back pain, which is particularly, bad. The problem here that most of the reasons why low back pain is evolving are not clear. So it's unspecific reason. That's kind of makes it more tricky. And I've tried to group, the problems causing low back pain into 4 different categories. So you see that tension or injury could be a reason. Then we have diseases, obviously, that could cause problems, movements or posture. Posture is something that can cause low back pain and structural damage, like osteoporosis or so. This is kind of obvious. But interestingly, not only we here on Earth are suffering under low back pain, also astronauts in space are suffering under low back pain. And this is a graph you've shown before, so it's quite the standard graph of showing what's going on when our body is exposed to microgravity. I just added low back pain to it because half of all the astronauts will suffer under low back pain. And this is something that appears within the first 2 days. Obviously, low back pain is not a life-threatening situation. However, it's bothering the astronaut, especially when they want to sleep. And this constant pain in the lower back, then eventually also reduces the quality of the work and in general, and especially the sleep quality is reduced. So interestingly, when the astronauts are going in this fetal position, like, bending the body and bringing the legs up to the chest helps reducing the pain. And that could be one hint what's the reason of low back pain behind. So low back pain is often related to degeneration of the IVD, the intervertebral disc. And this is also something, the IVD, that degenerates over the age. So by 50 years, most of us have degenerated IVDs. And aging is quite crucial for that, but also, genetics, smoking, diabetes, and all the other things. So that can contribute to low back pain. And here you have an overview what's causing then the pain. It's actually one part of the IVD is then pushed outside and can then touch the nerve, and that causes then eventually the pain. Down here you have the picture of a healthy and a damaged or degenerated IVD, and you can clearly see that in this damaged part here, we have that part of the IVD that pushes on the nerve, and that's quite obvious that you have problems here. So far, the treatments, because it's not clear and kind of difficult to handle, you just try to be active as possible, as much as possible. That's one thing. Or, and you have like painkillers you're taking, simply like that. And the final situation is then when you actually remove the IVDs and you actually fuse the thing. This is something that we're working on it, so it has a relevance for the people here on Earth, but especially also for the astronauts because they have similar things. Okay, this is kind of an overview on what's going on with the IVDs itself, so how this degeneration process is taking part. So again, we have the IVD, and composed of three particular regions. We have the nucleus pulposus, annulus fibrosus, and we have the end plate around. And so in a healthy state, you see that we have three compartments clearly distinguishable, and there are no nerves and blood vessel ingrowing. So that's a situation. When degeneration starts, quite often we have the extracellular matrix that degenerates together with inflammatory oxidative damages, that structural integrity of the IVDs is also reduced, which then causes a dehydration of the nucleus. Dehydration means that less water is in it, so the IVDs collapse and are not able anymore to withstand the pressure that is coming from the upper body. And so with that additional pressure that cannot be taken up by the IVDs, other structure of the IVDs are then stressed in a higher amount, like the annulus fibrosus around, that then eventually cracks and breaks and causes the problem. So this is the situation of a damaged IVD. So we have, no clear compartments anymore, and we have ingrowth of nerves and blood vessels, things like that. So that's actually the situation. What we're going to do, or what we're doing at the moment, is actually approaching the problem on various levels. So we're focusing on motor control. So is there a particular movement of astronauts that causes, particular stress to the IVDs? So we're looking at that. Then we have studies using ultrasound to focus on the 3D shape of the IVDs. And then we're doing histological and cytological analysis of the tissue itself. So we have an animal model that's going on. So you see that we have the human body, the astronaut that we're looking at, but we have in parallel this animal model that we're focusing on intra-tissue or cellular processes that we can focus on. So here, what we're doing with the body itself, so we're looking at the tension of the body. So we're looking at the stiffness, the stiffness here. So we have this device here, and we measure the force that the muscles is giving us under various situations. And interestingly, when we're doing this, also in parabolic flight campaigns, we've seen that the stiffness is actually reduced under higher G load, and is enhanced under microgravity conditions. So when you're in microgravity, it's kind of interesting, when you're just free floating, your back is stiffer than under normal conditions. So that was kind of interesting results that we gathered by the first studies. Now, I said we also look at the movement of the astronauts and the test subject, of course. So from that movement, we can calculate what force is on the vertebra, on the IVDs in between. And as you can see here on this graph, there is actually quite a dramatic increase of the force after the flight. But, on the very low part of the spine. So this really points that it's just the lower part that is especially touched by the situation. When we look at the curvature, we see that after the flight, quite often we have astronauts with a higher lordosis. So really the upper body is kind of collapsing because the muscle, most likely the muscle strength, reduced. And this increased lordosis can cause then the low back pain. Here, this is an astronaut without low back pain. And in that case here, we have an astronaut with low back pain. So that really determines or shows that this could be the case. Then ultrasound, I said, we're interested in what's going on with the IVDs. Under microgravity, they can actually grow because there's no pressure from the gravity on the top of our body. So the IVDs take up water, they increase, and that we're trying to measure by using ultrasound. However, this is quite tricky, and this is an ideal version of the readings that we got. So it's not always like that. Anyway, so this is the part that we used, with the humans in general, but now we're using the data that we're gathering, the extra force that is on the vertebra and things like that, to, do animal studies with similar situation. And for that, we built this IVD bioreactor where we can create compression, flexion, and torsion on the IVDs. The IVDs sit then inside chamber here, the heated bioreactor cultivation chamber. These IVDs are flushed periodically, so we can expose these IVDs to particular situation over several days, weeks, and theoretically months. The whole thing is mounted on the RPM, so we can even simulate microgravity. This is, you see here the deflection mode, just as a demonstration how the whole thing is. And because it goes on the RPM, we can simulate all the things. So trying to come as close as possible to the situation that we have in the astronauts. This is the approach that we're following with the IVDs. So yeah, we were using the bioreactor to simulate the situation. Then we analyze it on various markers, extracellular markers, or inflammatory markers that we're looking for. We go for the histological appearance of the material before and after the exposure on the IVD bioreactor. And this is quite interesting. There are progenitor cells available in the nucleus pulposus, so potential stem cells. And studies have shown that these residential cells sitting there in the nucleus can be activated or can be motivated or whatever, when you apply a certain mechanical stress or stimulation to the cells. And so this is our approach that we're trying to actually enhance or stimulate these particular Tie2 positive cells, which are progenitor cells, stem cells in the nucleus pulposus, to then initiate a self-regeneration, self-healing process. That would be ideal, Instead of taking, painkillers, you could do particular movements, stress the cells in a way that these cells, these stem cells, are motivated to get activated. So Yeah, and with that, we have that's kind of an approach, quite a regular approach, isolating cells and then focusing on these progenitor cells. Here we're using a particular approach like impedance cell sorting, so we don't have to label the cells anymore. So we're using electrical characteristics of these particular progenitor cells to sort them. So it's a nice and unique way how to grab the cells, the stem cells you want, and then, enhance them and pet them so that you can eventually bring them back into the body. And this is the standard, we do the data analyzing via mapping, and this works quite well here. I selected some genes we tested on type 2 positive, type 2 negative, and the rest of the cells. So the thing is working. Yeah, and at the end, it's basically, on one side, we would like to help people here on Earth with the data we're gathering from the research we're doing. So learning more about the degenerative process, obviously, finding regenerative strategies to enhance this health healing process, and actually to find new ways so that a replacement of the IVDs is not necessary anymore. On the other side, the astronaut, of course, we want to find out what is the reason for this low back pain in astronauts, so gathering more data. Then finding out about the mechanism, the biochemical or cellular mechanism that is involved, and actually then offer the astronauts some treatments, how they can overcome the low back pain. All right, so this is the approach we're now running. It actually helps that we're targeting a problem of astronauts that has a high relevance here on Earth for the patients. And so that also helps to find fundings and things like that. So this cross thing is quite good. And also the approach actually helped a lot that we're using humans. And in addition, the cell biology, the animal model that we're doing on bioreactor. All right, thank you very much. Thank you for your attention.
Ladies and gentlemen, I failed to inform you the format of today's session. We're not going to go for questions straight away, so I hope that you remember them or write them down. You all got an ESA notebook the other day, so you have no excuse. So next up, who do we have? Niels. There we go. So Niels is from Leipzig University, and he's going to talk to us about the radiobiological characteristics of multipotent mesenchymal stromal cells. Thank you, Niels.
Thank you very much. Thank you. So to put it in Monty Python terms now for something completely different, my name is Niels Nicolai. I'm a radiation oncologist by training, and I'm going to present some work that was done in our lab by 2 doctoral students. And my field of research is to target radiation-induced tissue damage as occurs during cancer treatments. And this is basically a byproduct that I'm happy to present today. No? Well, okay, thank you. So you have all heard about space radiation during some of the previous sessions, and as you all know, there are different types of space radiation: cosmic and extra, galactic and extragalactic cosmic radiation, solar particle events, and so on. And these usually high linear energy transfer radiation types can have serious side effects. The most worried side effect is the acute radiation syndrome that occurs with very high doses at a short time. But as we also know from cancer treatments, there are also other types of radiation-induced tissue damages that occur much later and at a later and lower likelihood. There is an increased cancer risk, obviously. Sorry. There is a time delay in switching. Okay. There is an elevated risk of cardiovascular disease that we know also from breast cancer treatment, for example, and there is an increased risk for neurodegenerative diseases. Our lab has been focusing on a certain type of adult stem cell, the mesenchymal stromal cells, that may provide a cell-based approach of addressing radiation-induced tissue damage. We know that these cells can migrate towards damaged tissues. They can create a protective or regenerative microenvironment, and they can also differentiate into functional tissue cells. Mesenchymal stromal cells, for those of you who do not know these cells, they form a very heterogeneous population of adult stem cells. They can be found in various tissues, not only the bone marrow, but also several organs. And these cells are quite tricky to characterize because the characterization does not only require molecular markers, especially surface marker expressions, but also functional characteristics, especially the ability to differentiate along different lineages. And we know from the cancer treatment area that mesenchymal stromal cells can address tissue damage induced especially by chemotherapeutic treatment. They can reduce apoptosis in normal tissues. They can produce an immunomodulatory microenvironment, immunosuppressive microenvironment, and they can protect the function of several organs, especially in the field of cancer treatment important. Sorry, I seem to press always the wrong buttons. The liver, the bone marrow, the bones, and so on. And we know from previous publications that these mesenchymal stem cells can be carried to space. There are two publications out there that have tried or have succeeded to culture mesenchymal stromal cells on the International Space Station and compared those cultured cells to those cells that were cultured on Earth. And basically, both publications suggest that it's possible to culture those cells in space. There is no significant effect on the molecular patterns of these cells, and there is no significant effect on these multilineage differentiation effects, so the functional capabilities of these cells. But if we want to pursue mesenchymal stromal cells as an approach to treat radiation-induced damage in space, especially for prolonged manned space missions, there is one link missing. Basically, the question, what is going to happen to these cells themselves when they are exposed to high LET radiation? And this is something that I'm going to show you. We obtained human mesenchymal stromal cells from the bone marrow of healthy donors, and we used an established radiation-resistant fibroblast cell line, the HS68 cell line, to compare the radiation effects of this resistant cell line with the radiation effects of human mesenchymal stromal cells. We used different types of particle irradiation, protons, helium ions, oxygen ions, and carbon ions. We stopped at oxygen ions because all these ions were produced at a cancer treatment facility that could not use higher energy or higher weight ions. So, but in total, this is probably the vast majority of particles that is used or that occurs during space radiation events. And first, we investigated the so-called relative biological effectiveness of these radiation types on mesenchymal stromal cells and compared those to the established resistant cell. And as you see, the range of the relative biological effectiveness is quite comparable. It's slightly higher for the heavier ions in the mesenchymal stromal cell preparations, but in total, there is no significant deviation from established RBE values. The morphology of mesenchymal stromal cells does not change during higher doses of ionizing radiation, also with particles, and we used biologically equivalent doses for the different particle types, and there is no morphological sign for apoptosis or cell swelling, which may suggest that these cells get unhealthy after exposure to ion radiation. We investigated also the molecular markers and molecular patterns of these cells, and as also suggested from space culturing of these cells, exposure to particle radiation, high LET radiation, does not change the pattern of the defining molecular markers of mesenchymal stromal cells. And the key issue, obviously, is the functionality of these cells after exposure to space radiation. If we want to use those cells as a cell-based treatment for radiation-induced tissue damage, we have to make sure that the functionalities of these cells remain intact. And we looked into the induced multilineage differentiation potential of mesenchymal stromal cells by inducing differentiation along the adipogenic, the chondrogenic, and the osteogenic lineages. So basically differentiating mesenchymal stromal cells into fat tissue cells, cartilage cells, or bone cells. And we were still able to do this differentiation after exposure to moderate doses of particle radiation at a level that is expected during moderate solar particle events, so also higher radiation according to the levels that we can observe or have to expect during longer manned space missions. So there was no abrogation and not even reduction in the potential to undergo induced differentiation along the individual lineages. Also, space radiation did not induce apoptosis, cell death in those cells. There was a slight increase, obviously, but no general or no massive increase in induced apoptosis after high doses or higher doses of particle radiation. And these cells seem to be quite resistant to apoptosis induction, and in order to see why that may happen, we also investigated the ability to repair the damage that is caused by ionizing radiation in these cells. And we looked at the phosphorylation of a histone variant, the so-called gamma-H2AX, that forms clusters or foci at the sites of DNA double-strand breaks. And as you can see in these immunofluorescence images, there is quite a strong increase in these gamma-H2AX foci 2 hours after exposure to not only photon radiation that we used here as a control, but also all types of particle radiation. And as you see, the foci become bigger with the heavier ions, and there is a lot more clustered DNA damage that is also more difficult to repair for these cells. But when we then looked at the dynamics of these gamma-H2AX foci, we could see that most of these foci were gone by 24 hours after exposure to radiation. So basically, the DNA double-strand breaks were no longer present. And we also investigated the activation of certain DNA damage sensing proteins by Western blots. And we looked at the phosphorylation of the ATM protein, the phosphorylation of the downstream CHEK2 protein, the presence of P21. And as you see, basically, after 24 hours, the activation of these DNA damage sensing and repair proteins was basically returned to baseline levels. So there is a very effective repair of severe DNA damage, DNA double-strand breaks in mesenchymal stromal cells after exposure to ionizing radiation with particles. We published some of these data already, and you're most welcome to download the publication if you're interested using the QR code. But to sum up these preliminary findings, we found that there is a strong preservation of the functionalities of mesenchymal stromal cells after exposure to moderate doses of particle radiation, especially a big preservation of the multilineage differentiation potential and no alterations in the expression of defining surface markers. The RBLE values range in the range of other radioresistant cells. We found low apoptosis rates and an effective DNA double-strand break repair after particle radiation. So at that point, at least in vitro, and these are only preliminary in vitro data, we can assume that these cells show relative resistance to high LET particle radiation. Currently, the next step is ongoing, and I just want to show you very shortly what's the next step, or what are the next steps. We are currently in the process of using mesenchymal stromal cells, combining them with bone marrow cells in 2D and 3D co-culture systems to see if after particle radiation we can still observe a radiation protective effects by using mesenchymal stromal cells in this setting. And as you see from some preliminary data, ionizing radiation with particles induces syncytial connections between mesenchymal stromal cells and hematopoietic progenitor cells by expression or increased expression of N-catenin, beta-catenin, and other proteins as well. And the next step will be then to go into animal studies to see whether this effect also holds true in more complex systems. With this, I'd like to thank ESA for funding some of our research by providing us with the beam time, which is a critical issue. Oh, batteries are running low. And I'd like to thank you very much for your attention.
Thank you very much. The slide changer didn't have its coffee break, so we'll just wait for it to be replenished. Next up we have Paolo Annibale from St Andrews in the UK, investigating effects of altered gravity on, well, do you want to introduce yourself perhaps? That would make a change. Go ahead.
Yeah, hi. Thank you very much for the invitation. As Jan mentioned, I've been working since 3 years now at the University of St Andrews in Scotland. However, the project I'm showing originates about 4 years ago, thanks to some repurposed COVID funds from the DFG in Germany, where I was working at the time. So it was all a bit serendipitous. And then ESA kindly stepped in and helped us to continue the project as I moved to the UK. So, okay, I don't know why it's animated. So the technical title of my presentation is the study of the effects of altered gravity on adrenergic-mediated cyclic AMP signaling in intact cells. So this may not relate with many of you. So the more relatable title, maybe also a bit more ambitious, is illuminating how individual cells respond to drugs in micro and hypergravity. Okay, so the story, in my opinion, starts a bit short of 40 years ago with a mission. It was the 7th mission of the Challenger, when the payload was Spacelab 3. So Spacelab 3 was a science mission from the European Space Agency. Remarkably, I found no historical information on the ESA website, so the only information I found comes from these NASA web pages. But basically, the Spacelab was a microgravity research lab, which was meant to conduct a number of experiments in different areas, including some studies of the effect of weightlessness, so microgravity, on animals. And in particular, there were 12 rats on board, paired to corresponding controls on the ground, which, upon return of the mission, were sadly for them sacrificed. And one of the goals of the mission was to study the morphology and biochemistry of the heart muscle upon prolonged about 7 days of microgravity exposure. So I think all the presentations of the previous days and also earlier today have introduced the notion that there are significant cardiovascular effect and deconditioning in microgravity. So that was the rationale for doing these experiments. And the main results that I'm showing here, just qualitatively, came, in my opinion, from a group of Delmet Philpott, who at the time was the head of the electron microscopy at NASA. So he was studying everything from rock to to mitochondria of animals. And they also did some biochemistry in this 1987 work. And interestingly, they focused on two enzymes, this one called phosphodiesterase and this one called protein kinase A, in order to study whether those were altered in the heart tissue from these rats. And the results were a bit of a mixed bag. So they acknowledged that they weren't fully consistent, nor with the hypothesis, nor some of the results amongst each other. But a few years later, they basically summarized the story by saying that, in their opinion, these protein kinases, cyclic AMP protein kinases, and we'll come to this in a second, in the left ventricle are compromised, suggesting that basically downstream effect will be altered. Okay. So why this was interesting for me? Because effectively what they did in this study was dissecting some of the molecular actors in a crucial signaling pathway, which is the adrenergic cyclic AMP PKA axis. Now, if I try to scare you now somehow, so what will happen, or one of the things that will happen, besides probably you laughing back at me, will be that your heart rate might increase. And this is mediated by the fact that your adrenal gland will release an hormone called epinephrine or norepinephrine, or adrenaline, if you wish, which will go in the bloodstream and then bind, amongst other things, to receptors in your cardiac muscle cells, cardiomyocytes, called beta-adrenergic receptors. And once it binds to beta-adrenergic receptor, there is a signaling cascade of events, which includes the production of an important molecule, which is like calcium, a currency that the cells uses to do a lot of things, called cyclic AMP, shorthand for cyclic adenosine monophosphate, which in turn triggers the activation of this protein kinase A, which you saw in the previous slide, which in turn basically regulates and controls calcium handling, and so cytosolic concentration of calcium are elevated, which allows enhanced contractility. So this leads to the chronotropic response and the increased heartbeat that we're all familiar with when we are under stress or anxiety. Okay, so, but what is more relevant, in my opinion, is that these beta-adrenergic receptors are basically prototypical members of a largest family of receptors called G-protein coupled receptors. So they all signal in a very similar way. I mean, they don't exactly signal in a similar way, but one common mechanism to this family of signaling proteins is basically to control the cytosolic level of this cyclic AMP protein, which is, together with calcium, I repeat, a crucial second messenger in the cell. And the other interesting aspect is G-protein coupled receptor effectively are the mediators of a number of extracellular stimuli. So they convert extracellular stimuli down to photons, because rhodopsin in your eye is a G-protein coupled receptor, into an intracellular signaling cascade. And now the interesting part is that, and that's what for me struck me as important, is that these are basically the target, a very important drug development target, and about a third of approved drugs from the FDA, but assume the numbers are the same in Europe, are aimed at these G-protein coupled receptors. So if you were to open the medicine pouch of an astronaut flying to Mars in a few years, a third of the drugs statistically will be targeting these receptors. So the question is if this pathway is affected, how is this affecting our ability to treat astronauts with drugs? So how is the pharmacology of these G-protein coupled receptors affected in altered gravity? And surprisingly, after these initial experiments in the late 80s, there wasn't much follow-up on this that I could find. So there were a few follow-up studies that look specifically in the heart at the effect of microgravity, and probably unsurprisingly, I'm sorry for the poor resolution here, or something happened in the conversion. So, unsurprisingly, so some of the results are in complete contradiction between each other. So in some cases, you see that the response to these drugs against this receptor that stimulate the activity of this receptor, which are called agonists, is enhanced in microgravity. This was a bed rest study on humans, and something similar was observed in parabolic flights, although here the things are more complex because we alternate micro and hypergravity. But at the same time, when people look specifically at another receptor of this family, which controls the level of this cyclic AMP in the cells, the observation was the opposite. So they saw that actually the level of this molecule were going down in microgravity conditions compared to the control. So effectively, and maybe without too much originality, what we did here was to basically try to bring new tools to this rather old problem. And new tools, I'm a microscopist by training, so new tools are basically imaging. So the question is, how do you do biochemistry? By imaging, so one way is to use biosensors, fluorescence biosensors. So what you see here is a schematics that shows a biosensor for cyclic AMP. So effectively, the biosensor is a protein attached to two other fluorescent proteins. So these are all genetically encoded, so you need to feed them to the cells. And these two proteins talk to each other via a mechanism called resonant energy transfer. So effectively, if they are very close, the cyan protein cannot fluoresce because all the energy is stolen by the yellow protein. But if cyclic AMP binds, this domain opens up, so there is a structural modification of the protein. They get farther away, so the cyan protein is now free or unquenched, and it can emit fluorescence on its own. So effectively, you need to image in two colors, the cyan color and the yellow color here. You see one is called the donor channel, the other one is called the FRET channel. And if you give a stimulus, which increases cyclic AMP levels in the cell, you see that these cells, these are HEC293 cells, which are basically a workhorse in single-cell pharmacology studies. The cyan signal goes up, whereas the yellow signal goes down. Okay, so just reports. We have quite some experience in the last few years working with this biosensor, so it seems like an obvious thing to adapt this to this setting. The other tool is basically the ability to do these measurements in altered gravity conditions. And here what kicks in, I'm sure you will hear more in a session from Christian later today, is basically the facilities as the DLR in Cologne, where we have the ability not only to subject cells to altered gravity, but also to image them, in this case in hypergravity, while conducting live cell microscopy. I saw some pictures in previous talks about the centrifuge. So that was with a bed for people. So if you kick out the bed, you can mount a microscope called the hyperscope and do your microscopy. So what came out over this last about 2 years of experiments that we did over there? So the first observation is that if you do experiments in hypergravity, again, what I'm showing you is what is called a concentration response curve. So just to explain what this means, so what you see here on this axis is basically response of the cells to the stimulus, the drug against the beta receptors. So if you go down, you have more response. Apologies, this is a bit counterintuitive, but it depends on the way you calculate these fluorescence ratios. And so if you have basically very little drug, this is a molar concentration in logarithmic units, so nothing happens. But after a certain amount of time, the receptors basically switch on, and you have a response which increases the level of cyclic AMP in the cell until you have basically saturated all the receptors and nothing happens. So the blue is the 1G control, and the red is the hypergravity. We use 2G to match some of these earlier data from the 87 work. And what you see here, the red, is basically the hypergravity data. And you see a slight shift to the left, which becomes more prominent if you keep the cells going for longer, like 8 hours, and then it's very clear up to 24 hours. So what does this mean effectively? This means that if you give a certain concentration of drug, you have a much larger response for the same concentration in the cells that have been exposed to hypergravity. Or if you turn this around, it means that to achieve the same response in the hypergravity cells, you need about 10 times less drug. Okay. Now, if you do this and you monitor the cells live, you can then do movies, and you can see again here the cyan gets cyaner, the yellow gets less yellow, and you can basically calculate the kinetics of accumulation of cyclic AMP. So in this case, we were perfusing the cells with the drug with a peristaltic pump working on the centrifuge. And what you see here is the control, which basically displays a certain kinetic of accumulation of this. So this is effectively the kinetic of your response to the drug stimulus. And that's what happens in hypergravity. So here you switch on the centrifuge. There is a basal response, which could be a mechanosensitive response, but that's another story. And then you have but the interesting thing is that you have a much faster response in the generation of the second messenger in the hypergravity condition as opposed to the ground control, which is consistent with the data I just showed you before. Now, in microgravity, things are a little bit less. There used to be here error bars as shading, which didn't transfer again. So my apologies, but these are all replicate experiments done over hundreds of cells and 3 campaigns. So what you have to use is a clinostat. Here you can't image live, so you have to expose the cell to simulated microgravity by clinorotation. Then you have to fix them, and then you image individual cells. So we get a readout from each single cell. There are thousands of cells in each slide, tens of thousands over several slides. And then you can build again this concentration response curve. Interestingly, what happens in microgravity is the opposite of what you saw in hypergravity. So in microgravity, you have a shift of this concentration response curve to the right, which effectively means that to achieve the same effect, you need more drug. Okay? Then, okay, we said molecular, what is causing this? So what are the molecular determinants of this process? So if you do some survey of single cell imaging in altered gravity conditions, the thing that comes at you very obvious is that everybody seems to agree that there are alterations to the cellular cytoskeleton. And this was also fitting very well our hypothesis, because independent of all these microgravity studies, I mean, we know that these membrane receptors do have intracellular interaction with the cortical actin. So the actin under the cell surface affects the way, or is in the way, if you wish, of these membrane receptors, and there are specific interactions that mediate this process. So this was, okay, say, well, if there is a remodulation of the cortical actin, this could explain the change in function at the receptor level. And again, apologies, again, something didn't work here in the transfer of the data. But for example, if we alter, this is ground data. So if you, for example, increase the polymerization of the actin using a mutant, what you see is that you have a shift to the right of this concentration response curve. So the receptors tend to signal a little less well than if there is less actin. So this seemed like the golden key to understand these effects. However, when we started to actually look at the actin, and I throw this at the audience because I'm sure there are in the audience people that have been studying this way longer than me. So in a way, it's my question to the audience and my small problem that I'm throwing back at you. So if you look at the actin polymerization, these are HEC293 cells where actin has been labeled with LIFACT, EGFP. They've been fixed and imaged in confocal microscope. So this is just a small selection of hundreds of such snapshots. These are 1G controls, and these are 0G cells. Now, if you do this a lot of times, okay, what you will end up, your eye will tell you that not much is happening. And if we don't trust the eye, we went up to several approaches for quantitative characterization of the organization of the actin. One is, which we like the most, is based on basically calculating the orientation of actin filaments. So here you see the actin filaments color-coded according to their direction. We did this in collaboration with a group in Lausanne at TPFL. And if you do this analysis on the HEC293 cells, and you plot basically for each cell the average orientation of the actin filaments that you can detect. And you do this, for example, at 2 planes, the basolateral membrane, the one that sits on the glass, and in the middle of the cell. Again, the somehow sad conclusion is that you don't see much of a difference here. Say it could be the overexpression of the LIFACT. Well, we went to human pluripotent stem cells where actin was tagged by a knock-in of a GFP by CRISPR-Cas9. And again, if you look at this 1G versus 0G, I will make a very long story short, you don't seem to see much of a difference. So effectively, we have some very interesting observation about function. We see basically a different response to drugs in altered gravity conditions, which I personally think could be very relevant for a number of issues that we have seen over the last 3 days, but we don't have a mechanism. So what is next for us? Well, the first step will be to now bring actual pluripotent stem cell-derived cardiomyocytes to the facility at the DLR. We secured a transfer incubator recently with the cell box, and so these are some of the cardiomyocytes we are growing in St. Andrews. The other key thing is to basically generalize. We have been focusing on beta-adrenergic receptors because we like them very much for other reasons, but it's crucial to extend this to basically there are 800 G protein-coupled receptors, so we need to start moving horizontally to other of these receptors. The other thing that will be very interesting to do if this project can continue and continues to secure funding will be to basically look at the organization of the receptor at the plasma membrane. Again, there was a number of studies in the mid-90s using immunogold electron microscopy that looked at supramolecular organization of membrane receptors. There wasn't much of a follow-up, which again is remarkable considering that since now over a decade we have super resolution microscopy methods to look at this in a way which is definitely more precise and physiological than electron microscopy. And then two things that just came up, I think, during chats with colleagues over the last couple of days. So liquid-liquid phase separation, which was discussed in the panel about soft matter, is a process that is now overly invoked to explain changes in cellular signaling. And so if this is affecting altered gravity, it will actually be an important thing to know. And the other aspect will be the ability, obviously, all these measurements could be done on the ISS. You're all aware of the FLUMIAS, which, however, was delayed, so it won't fly until next year, 2 years, nobody knows. So it's still quite relevant to try to do some of these experiments in simulated microgravity on Earth. So what is somehow missing is the ability to do live cell imaging during microgravity exposure, because the centrifuge gives us hypergravity, but there is not yet an on-clinostat microscope to do live microgravity measurements. So, and with this, I would like to acknowledge all the people who over the last 2 years did work on this project. Also the team at the DLR who was basically invaluable to conduct this experiment, Christian and Yannick, Christiane from ESA who steered us through all the administrative aspects, and also this company, Cellbox Solution, that provided us with this transport incubator, which we tested about 6 months ago for the first time, and they also wrote a small user note about our applications in microgravity, and of course our funders, ESA, EHRE, and the DFG that provided these repurposed COVID funds that started this all. And thank you very much for your attention.
All right, we go from human biology to synthetic biology for space exploration. Claudia Pacelli comes to us from the Italian Space Agency. Thank you very much.
Thank you. Okay, good morning, everybody. First of all, I want to thank ESA for inviting me to talk about synthetic biology for space exploration, which is a topic that we discussed a lot in the frame of a topical team financed by ESA. I'm Claudia Pacelli, and I'm a researcher at the Italian Space Agency. And I would like to start with this picture that reminds us how far we have to go for our last, for now, destination, which is Mars. And without taking into account the time to go there, we need to remind that which kind of challenges our body and also the instrumentation, everything we bring with us, we will have to face when we arrive on the planet. So we will have basically very few amount of oxygen, very strong radiation environment. We have heard a lot in the past few days about that, and the gravity is approximately 38% of the terrestrial one, and we will have the presence of perchlorates on the surface, which can create troubles as well. So can really humanity develop sustainable and autonomous outposts beyond Earth? Here I reported a recent study that has calculate the total logistic mass that has been delivered to the International Space Station from 2017 to 2023, and it has been calculated that around 130 metric tons have been delivered to the ISS, and the 40% of them, more than 40% of them, are consumable material for human stay there. And what about on Mars? It has been calculated that for one crew member on a long duration mission of 30 months class or 30 months, and here I'm referring to the conjunction class, it has been calculated that we need the one crew member, we need more than 2,000 kilograms of water and more than 1,000 kilograms of food. And this is without taking into account the cost of course, of the exploration. So just to refresh you that to bring 400 grams of payload there, like a lemon soda can or a soccer ball, when it costs approximately $10,000. So the first trouble that we have is, of course, the up mass constraint, but that we have also other problem like the expiration date of the material that we bring with us, or I'm referring also to drugs. So how we can do? The big idea is to use synthetic biology for space exploration. So thanks to the development of bioinformatics, machine learning, artificial intelligence, development that we have in the in the past years, we of course we can implement synthetic biology in space. So the basic idea is, since we use on Earth for millennia biology to produce chemistry, we can do the same, of course, in space. And in this context, ESA in 2022 has established a space synthetic biology topical team that has been coordinated by Professor Silvano Nofri and Dr. Ralph Mueller, to who I would like to dedicate this talk. And there are a lot of member representatives that are also present here, like Daniela Billi and Natalie Leis, and we started to identify the key area where synthetic biology can help to sustain human presence in space. And of course, we think that synthetic biology can be useful for implement the in-situ resource utilization. It can implement bioregenerative life support system and food production, and also the strategy to protect human and microorganisms to stress and radiation, and of course, last but not least, human health. So the in-situ resource utilization, just a refresh, it's the processes of using material found directly on the planet on a specific location rather than bringing resources from Earth, and this, of course, can impact on the problem that I was talking before about the up mass constraint. So organisms, not only microorganisms, but also plant, can be used to feedstock production, material and chemical production, bioprinting, chemical production, and also biomining. How can synthetic biology implement the ESA-HRE? By enhances the capability of this organism to advance the technological readiness of the ESA-HRE. So here we are referring to cyanobacteria and algae. Of course, they are the primary autotrophic production organism that can be useful for feedstock production, but also the anaerobic acetogenic prokaryotes they use carbon dioxide and hydrogen to produce intermediate of the feedstock in bioprocesses, but also yeast and fungi, and they are really useful to degrade crop and water waste, recycling them back into food, and they can be food as well itself. So then we focus on the bioregenerative life support systems and food production. And again, we by engineering microorganisms and organisms, we can overcome the limitation associated with the wild type and introduce new capability. So in this way we can enhance the efficiency of metabolic pathway, the bioproduction, and also the robustness by using modifying organisms with new function, for example, resistant to radiation, resistant to dry, we can enable microorganisms to work in a robust way under the bioregenerative life support systems. And also, synthetic biology can make possible to overcome the challenges of agriculture in an extraterrestrial environment. Even in plants, we can fortify the plants. We can engineer the plants to produce more nutraceuticals that are really important to to support the astronaut health and, in general, strengthen the defense against space stressors. Of course, I was talking about protection from stress, so here I'm referring not only to radiation, but also to dust, toxic compound. What we can learn from resistant microorganisms is we can study their activated mechanism in order to identify genes or protein that can allow us to overcome this trouble. And also, microorganism-based research is also important to developing possible, let's say, organic composite materials, but also dietary supplements to contribute to the overall strategy protection from radiation. And human health, we have heard about a lot of these days how much trouble we have going to space, and one of the problems that we have is that we cannot have drugs that we can have drugs for all the mission, for example, to Mars. So we can use engineered microorganisms to produce drugs on demand, so the so-called astropharmacy, opioids, antibody, antivirals, vaccines, and so on. And in this way, synthetic biology can help by simplifying the complicated metabolic pathway for the bioproduction of drug and microbiome. It has been quite clear now that it's really important to maintain a balanced microbiome during the long human mission. We know that the impairment of microbiota is not only due to microgravity, but also for the isolation confinement condition, and the microbiome has also the power to interfere with the other physiological system. And here I'm not referring only to physiological system, but also it interferes with the cognition, for example. This is really important to guarantee the success of the mission. And synthetic biology can be used, of course, to create, modify microbiome in order to use non-pathogenic strain and to reduce of the risk of infection disease, also taking into account that the immune system is depleted in a space environment. And we also consider the fact that we can use engineered microorganisms to produce biosensor. And here I'm referring not only into incorporate engineered microorganisms into material patch that can directly release drugs or everything that the body needs on demand, but also we can also build environmental biosensors so that microorganisms can recognize if something changes in the environment or like radiation, but also chemical. So here I'm going to the recommendation of the topical team, which are, we consider the recommendation in a short, medium, and long term, of course. So we highly recommend the functional continue to do some functional studies, mainly on extremophile, because we can of course learn a lot by their strategies to adapt on stressor environment, and in order to identify new strategies to overcome the challenges that we will have in space, and to improve the general resistance of the compartment, the component of life support system, or ESA-HRE. And here I see very strong interconnection, of course, with astrobiology field. And yesterday we had very good session, getting the feeling of how much they are interconnected. And so we but we really need to identify and optimize the organism or the microbial community that can support function under space-relevant condition compared to the terrestrial one, because the major challenge is when we bring microorganisms in space, it's the fact that they have evolved on Earth. Of course, we know that they are able to withstand space stressor, but the majority of the study that has been performed so far has been performed in dry conditions, but as it has been highlighted yesterday in the astrobiology session, we need to move to the active experiment. So we need to know how they change, how they can adapt and live really in a active condition in a space environment. And so we recommend to use well-known prokaryotes like Bacillus subtilis and cyanobacteria that are known that it's quite easy to modify genetically. For Bacillus subtilis, we know that it's able to resist to space condition. Also, we have a lot on cyanobacteria study as well. And for yeast, use easy to grow safe yeast like Saccharomyces cerevisiae should be prioritized. And of course, in general, we need to test the impact of real space environment, radiation and microgravity on chassis robustness by using the omic approach, such as genomic, transcriptomics, and so on. And establish standard protocol equipment to use engineered microorganisms in a safe way. So again, understand whether they change in active condition in space is crucial for the safety. And in general, advanced bioprocesses capability under laboratory simulated condition to mature the technology to go further in space. And at the end, the laboratory production should be extended, of course, in laboratory facility. And we have heard this morning that some bioreactors are already being implemented also on the ISS. So these are the recommendations in the timeline of human space exploration. So we recommend to continue to do, of course, ground-based experiment, not only in the lab, but also in a simulation facility. But of course, it's important to implement experiment in the low Earth orbit and beyond. So we hope to that similar studies can be performed in the cislunar orbit and in the medium long term to test the technologies developed on the lunar surface to finally achieve the final destination to use that on Mars. Of course, we know that we all know that science and technology that we are implementing in this space have a lot of benefit and continue to have a lot of benefit for Earth, and these are can fit also some of the global goals that has been identified by United Nations, and also this topic of high industrial relevance as well, since by enabling industrial bioprocesses to operate under more extreme non-natural conditions can enhance the production in terms of rate, tighter yield, and reduce, of course, the process cost, and it will also foster the circular economy. Here, just reported some of the benefits for Earth that we have identified. So, implement water purification system, ameliorate bioremediation, sustainably produce bioenergy, drugs, and food, and reduction of pollution, improve ground cultivation, improve urban farming, and achieve a sustainable method to grow food, and this is especially important in remote area on Earth, to develop the next generation system to enhance radio protection strategies by also utilizing biomaterials, improve general crop resistance to high UV radiation, and for human health, the development of tailored medical therapies, which is really important for orphan medicine, and also we aim to allow medicine in remote and resource-limited location on Earth. I want to leave you with just a final message. If you want to bring synthetic microorganisms in space, of course, we need to take into account the concept of biosafety, which is a relatively unexplored area, but in the ongoing discussion that are we are hearing a lot about planetary protection. We need also to take account into the biosafety of bringing synthetic biology in space. The results, if you want to know more, the results, the output of this group had just been accepted in FPGA microgravity, and I hope to see the paper out in the next few weeks. And with this, I'm done. I thank you for your attention.
Ladies and gentlemen, panelists, if you'd please like to take a seat at the front. Thank you. All right, ladies and gentlemen, we have a microphone, which I'm going to just give to the audience. And whoever claims it, I'm going to give it to you, and you can pass it to somebody else when they have a question. We have discussed a lot today. We talk about simulated microgravity. We talk about all these different model systems. We talk about changes in the biology, which maybe only occurs in 50% of astronauts, Marcel, with regards to lower back pain. Starting with that, how do we know who is going to be affected? Like, do we have biomarkers that can be detected, for example, in blood? Is this something, or is this are these markers through, do we need to detect it by ultrasound, for example, beforehand? What might we need in orbit or in deep space? And more importantly, when it happens, what are the challenges and potential solutions or countermeasures for those space missions?
Okay, it's working. Yeah, actually, it's a good question, and it's tricky to answer. To find markers or so that, in this example, that would determine whether a person would develop low back pain or not. So the important thing is to actually learn more about the causes. So what's the reason for developing low back pain, and eventually. Also internal, when we look at the muscles and psoas muscle and things like that, there are slim changes between the individuals. And finding out more about the mechanism that's causing these problems, we may relate to particular anatomical changes or differences between the individuals, and that could be a nice determination of whether you would develop a low back pain or not. So, but basically, we need to learn more about the cause.
Mm-hmm. So you're using, you have a model system, which you have in an apparatus. So you're using the bovine spine. Is that correct? Do you see a role for other three-dimensional biological models? And this also applies to those that have studied cells using two-dimensional monolayers. Like, do you see a role for three-dimensional models in understanding and mitigating these pathologies? To potentially also minimize animal use, because that is a program through ESA to minimize animal use. So how do you see that going forwards?
Yeah, it turned out that actually RPM or other devices to simulate microgravity can be quite useful, and that to grow cells or to study the development of cells and things like that. So that is really a benefit. However, you always have to be aware of the limitation. Like, for example, RPM, it's not like turning in all the directions slowly. It has to be adjusted and proven that it really minimizes, the gravity vector or so. So I think it's very useful to use all these tools, but you always keep in mind the restrictions and the problems that could evolve by that. So not just blindly trusting the results you're getting when you're using these devices. So be aware of the limitations.
Paolo, you were measuring the fluorescence changes, and these are in a two-dimensional culture. Are you expecting those changes to be occurring in a three-dimensional environment in the same way? How are you going to measure them? Or more to the point, what technology do you need to be able to measure those?
Well, I mean, if actually one of the next steps we have suggested for a continuation of the project is to move to three-dimensional cultures. So yes, I agree, it's important. If we work after exposure, and if you use any microscopy method that has a sectioning capability, 3D cultures, as long as they're not like 10 meters thick, will be manageable. For live imaging, I also think that if you're talking of organoids or things like this, it should be feasible to work easily and to conduct imaging in 3D cultures. I mean, we already have some cues that things change outside microgravity, so in 1G, between 2D and 3D situations, but some of the fundamental mechanisms, in my opinion, aren't gonna change. Okay.
And so, out of interest, what are the broader implications of those signaling changes? And more to the point, so in an astronaut, for example, what are the downstream effects? Do you expect similar changes to those that you see in your model systems?
Yeah, obviously,
and how to measure those, because you're not going to be able to image an astronaut. So
obviously, what we have been doing is pharmacokinetics in a single cell, whereas pharmacokinetics should be done in a person or is normally done on an animal. So we are clearly a few steps upstream of what you would observe in a full organism. But in my opinion, it's quite thought-provoking, because if you had the same changes at the organismal level, and these were, I don't know, paracetamol, the drug we were talking, we will be seeing difference like giving 500 milligrams versus 5 grams to achieve the same effect. So it's hard to speculate what will happen at the whole body level in the astronaut, but if we see these changes at the single cell level, to me this is a strong motivation to look into this further.
Ladies and gentlemen, do we have any questions in the audience? Did the microphone go roaming? It is now. Thank you.
Thank you. Well, thank you all for the wonderful presentations. My question is for Niels. Well, my name is Carmen Blanco, and I work in the field of biosensors. I'm professor of analytical chemistry, so I came across to this field of synthetic biology through some extracellular vesicles. I was wondering if in your studies you are exploring also the possibility of use extracellular vesicles as a way to respond to the treatments or the different conditions that you are trying.
Thank you very much. I think this is a very important point, and we know in the context of mesenchymal stromal cells, extracellular vesicles play a crucial role. We have not yet started to do that, but it's certainly on the agenda, and I think that may be a good way to further this project. Yes.
I would love to talk to you later then to discuss
Please
about these biosensors.
Great.
Okay, thank you.
Thank you, very interesting presentations. I want to comment to Marcel's presentation about when you ask if there is a 3D models which could be associated with spinal pain. I think one of the, I've come to my mind because Marcel showed that it is ingrowth of nerve fibers into the disc. And there are different type of fibers which can be. Some of them could be from pain neurons, which detect pain, and some can be just from motor neurons, for example. So it's very interesting to detect which type of nerves actually grow in, and if they really, the reason for this pain, And this can be, of course, done on 3D printing model. And also very interesting, you said that you're expecting that the result of this is a compression of disks, but it means that pain in the cosmonaut must be different reason, yes, because there is no disk suppression. So it must be this type of pain after they come back to Earth. Is it correct?
Yeah, actually in space, because there is no compression from gravity, you know, the IVDs are taking up water. This is the mechanism to actually withstand, to build the counter force to keep the body upright. So, and in space, because there is no force pushing back, they're just taking up water, they swell. That's the general idea. And this swelling, you can imagine that this is not a healthy environment for the cells in general, you know. And that may be a big reason or a key reason why we see degeneration in these discs then. And as you have pointed out quite clearly and correctly, you know, when they're coming back, these astronauts, you know, then this balance is totally disrupted. And, you know, the pressure that is then again on the IVDs can probably not be restricted, you know, worked against, and that could cause then the problem that the risk of developing a disc hernia of astronauts is actually increased by about 20% when they're actually returning to Earth.
Do you know if they develop neuropathic pain after? Is it any evidence that they increased risk of neuropathic pain after? Or it's just inflammatory pain which then goes away?
Yeah, hard to tell. I can't really say. Yeah.
Yeah, thank you very much for your answer. And also would like to comment, it's a very cool study with all these fluorescent proteins. It seems like with actin you don't see the difference even with all this analysis, very cool analysis you do. Did you consider to concentrate on the protein which go into the nuclear and from the nuclear, like YAPTAZ, for example?
Yeah, no, we haven't looked at any, you mean downstream signaling in terms of transcription factors. I mean, there are cyclic AMP, so if that was your question, so there are cyclic AMP-dependent transcription factor, like CRE, but we didn't look at all into that.
Because we looked at YAPTAZ.
Yes.
It's very clear dislocation from the nuclear, and you can see it with antibodies very similar to actin because they are associated with actin staining.
Right, but the question for us was more if the morphology and ultrastructure of the cortical actin, which is the one which is mechanically interacting with this membrane receptor, was changing. And the problem that I found in surveying the literature is that there isn't basically, so each paper deals with a different cell line. So there is no way to have a comparative study or to even relate to previous measurements. So it may be that we just picked the wrong cell type. The one thing that I found is that there was this recent experiment by NASA on iPSC-derived cardiomyocytes, and they looked at alpha-actinin, which is another marker of basically sarcomer structure in these heart cells, and they also didn't see any modification between ground control and 1G. So at the moment, my gut feeling is that maybe there isn't that big change in actin structure, but there might be definitely other proteins that, for example, mediate the interaction between actin and the receptor that are either up or down regulated, and obviously, but like at the moment it's like a fishing expedition, so we don't know where to look.
Hello, and thank you for interesting presentations. I come from the industry side, and we have this engineering approach for this space exploration. So quite a lot of new things for me on these topics. And I just would like to comment on these research facilities side and the needs from these scientists that we are currently developing this mini fluorescence microscope, which is planned to be used for this live cell imaging in space, and it's also possible to use that on these RPM machines for the microgravity research on ground. So it just would be very, very interesting.
Maybe we have a chat.
Yeah, to discuss those later on.
Yes, this is a question about mesenchymal cells, because as far as I know, the transition from epithelial to mesenchymal cells is a first step of cancer. So my question is, so this is what makes these mesenchymal cells stronger, and they can migrate, and maybe also stronger to against radiation. So my question is it safe to use to cure cancer mesenchymal cells, which are one intermediate step leading to cancer, as far as I know? Thank you.
Obviously, if you use any external cell in the body, as we know from hematological malignancies, there's always a risk of inducing diseases. I think the mesenchymal stromal cells that are abundantly available in healthy bodies in each of us, and the epithelial to mesenchymal transition, it's not the same, but nevertheless, there is a unknown risk of these cells of furthering the protection of mutated cells. And this is a key issue, obviously, and this is something that we and other groups are also currently looking into. On the other hand, you can buy mesenchymal stromal cell preparations, and there is a license in Europe to use these cells for the treatment of Crohn's disease, for example. And there is a big trial from the Netherlands where they used mesenchymal stromal cells, allogeneic mesenchymal stromal cells, to treat salivary gland dysfunction after radiotherapy for head and neck cancers. So, and in these both of these settings, there is no evidence of using mesenchymal stromal cells giving patients secondary cancers or bringing back the primary cancer that has been treated in the people in the salivary gland trial.
Ladies and gentlemen, I recognize that lunch is nearly upon us. In fact, it's now. Are there any further questions from the audience? Thank you. If not, two questions. I would like to ask Claudia, actually. Do you see a role for synthetic biology in supplementing human biology? And this comes to, for example, you mentioned that you could have a patch on the skin to release. What about individual implants that are organ and tissue specific that could respond to a changed environment in, like, for example, spinal decompression, you know, that could then activate and come up with a localized countermeasure?
Yeah, that will be amazing, of course, but I think that we are far away from this, so really we recommend to start to study from the laboratory to space implementation, and yeah, but I would love to have that, yeah.
Okay.
Thank you.
And I want to ask, you're going to say money, what would you need from ESA to expedite your research? Other than a lot of money. Okay. In which case, thank you. Thank you. In which case, thank you all for staying until 12:30. Enjoy your lunch. Thank you to the panelists and the speakers today. You'll see me again this afternoon. I'm taking over from Christiane Hahn in the 3D tissue talks. Thank you very much. Thank you very much.